Hello everyone and welcome back to Biology with Mrs. Evans. Today we are continuing our discussion on cell division. So in the last video we talked about the timing of the cell cycle, and we also talked about the first part of the cell cycle, which was interphase, and we briefly talked about mitosis. So today we're going to look at mitosis in more detail. Mitosis, remember, is simply the division of the nucleus, and eukaryotes, remember, it is a complicated process because DNA, as we've talked about in Unit 6, is double-stranded. It is too big to fit through the nuclear membrane, so there are basically things that need to happen in the cell in order for a cell to divide. And the 4 stages of mitosis is, as you see here on the slide, prophase, metaphase, anaphase, and telophase, or telophase. You hear it pronounced either way. And PMAT is basically the acronym for the 4 stages of mitosis. So the first stage of mitosis is prophase. So during this phase, the nuclear membrane or nuclear envelope, you'll hear called lots of different things, it is going to break down, because in order to move basically the DNA has to be able to move around freely, so the nuclear membrane is going to break down. The mitotic spindle for animal cells is going to form. So we talked about centrioles in Unit 4, and we said that they are responsible for the formation of the mitotic spindle and how the chromosomes are going to move. And in this video, you're going to see exactly how that works. And then also, the DNA basically starts to condense. It's going to become visible for the first time during prophase. So DNA has basically condensed to form the chromosomes that we see. Again, this is the first time that we're going to see DNA inside the cell. So the centrioles, remember, excuse me, are organelles that are found only in animal cells, and they are going to create a structure called the mitotic spindle. And I'll show you what that looks like here. So this is an example of the mitotic spindle. You can see the centrioles have basically moved to opposite ends of the cell, and basically they establish the poles, if you will, of the cell, and they create these spindles, which are basically made out of microfilaments and microtubules that are going to attach basically to the centromeres of the chromosomes, and they're going to be used to move the chromosomes around. So just like a fishing pole, as it starts to shorten those fibers, the chromosomes are going to be basically pulled towards the poles of the cell, and that's going to be used to separate basically the 2 sister chromatids, because remember, at this point the DNA has been replicated. So here is a typical cell in prophase. You'll notice that the nuclear membrane has started to break down. That's the indication by the little marks here. Notice that you see basically chromosomes for the first time, these little X-like structures. And then also the centrioles have moved basically to opposite ends of the cell. Remember, in interphase, the 2 centrioles were kind of hanging out here. It is at during this phase, prophase, where they're going to migrate to opposite ends and start to form the mitotic spindle. So now you would have basically fibers coming out and attaching to the chromosomes in order to be able to move them. All right, so that's prophase in a nutshell, and this is typically what prophase would look like in a cell. Metaphase is the easiest to remember because meta means middle. So during metaphase, the basically the centromeres will have basically been attached to the mitotic spindle, and they will have been the chromosomes themselves will have been moved to line up in the middle of the cell. So easiest one to identify, chromosomes are going to be located in the middle of the cell, and this is what it would look like if you were looking at it. So you can see that the chromosomes are clearly lined up in the middle, and they are basically getting ready for the next phase of mitosis, which is anaphase. So anaphase is the 3rd phase of mitosis, and the way that I remember is just simply remember A for away, or apart. And now what happens is the mitotic spindles, those fibers are going to start to shorten, and it's going to pull the two sister chromatids basically apart. And the sister chromatids are going to move towards the poles of the cell. So remember A for away or apart. And this is what it would look like. You can see that the sister chromatids have been separated. So now each cell is going to end up with a full set of basically the DNA that was in the original parent cell. So you can see this indentation starting to form during the late stages of anaphase, and it'll continue through basically telophase until and the last phase of mitosis is telophase or telophase. You'll hear it pronounced either way. During this phase, the chromosomes begin to relax back into their chromatin form. The two nuclear membranes start to reform around the DNA, and the spindle breaks down because it's no longer needed to move chromosomes around. So the spindle breaks down, and then the nuclear membranes reform around the DNA. And the way that I remember that is T for 2. 2 new nuclei have been formed from previous new cells are created. So this is what telophase basically looks like. You can see in the picture that the nuclear membrane is starting to reform, and you really haven't seen the chromosomes start to relax, but they will become no longer visible. And you will also see this is a cell that has started to undergo cytokinesis as well. And remember, cytokinesis is the division of the cytoplasm. So right at the end part of anaphase, cytokinesis will begin to start. Let me back up and let you see the. So after telophase, the last thing to do is officially divide the two cells into two, and that is done through cytokinesis. And it is different for whether it is a plant cell or an animal cell. So in animal cells, you know that animal cells do not have a cell wall. So what happens is basically animal cells have what's called a cleavage furrow. Basically the cell membrane is going to pinch inwards until it creates two new cells. And you can see here, once it completely pinches all the way through, you are going to end up with two new cells. So you need to know that animal cells do cytokinesis by the use of a cleavage furrow, and then plant cells, because they have basically a cell wall, they use what's called a cell plate. So you can see here's a picture versus an animal, an animal versus plant cell. So the cell membrane is going to pinch inwards, creating this cleavage furrow. And then a cell plate is going to basically start to grow in the middle of the cell. And what happens is the cell plate is going to continue to grow basically outwards until it completely cuts the cell out, and it becomes eventually this will become the new cell wall. So you will end up with two new cells. So you need to know the difference between animal and plant now that we're adding to our stuff that we've been talking about in Unit 4. Is cytokinesis is different for animals and plants, because in animals they have a cleavage furrow, and then in plants they use a cell plate. All right, and then this is again a review for the cell cycle. You've already seen this. We've already talked about interphase, which is the first part of the cell cycle, which is this. Remember I said most of the cells are going to spend 90% of their time in this phase. Quick review, this phase right here where the cell is growing is the G1 phase. This phase right here where the DNA has been replicated is the S phase. This phase right here is the G2 phase, and then we start to move into mitosis. And again, it's in order. So the first stage is prophase, followed by metaphase, anaphase, and telophase. All four of those stages make up mitosis, which again, remember, is the division of the nucleus, and then followed by that is cytokinesis. And really, it's not really followed, because cytokinesis is going to start basically towards the middle end of anaphase and continue through until the cell has completely divided into two. And both mitosis and cytokinesis are the two phases that make up the cell division piece when the cell is actually going to divide. So in the last video we talked about regulatory proteins, basically timing the process of when the cell divides. We talked about growth factor, external proteins that are telling the cell that it's time to grow. And then cyclins regulate the timing of the cell. So what happens if something goes wrong? Occasionally, the mechanism that basically makes sure that the cell timing is correct and everything, something happens to turn off that signal to basically stop cell division. And what that causes is cancer. Cancer, you need to know, is simply uncontrolled cell growth. So if for whatever reason the cells are going to continue to divide, and the cell masses that basically they create, as tumors. So cancer, uncontrolled division of the cell. And how do they treat cancer? Unfortunately, probably everybody that has seen or is watching this video has been impacted in some way, in shape or form, by cancer. Either a loved one, a friend has had cancer, and you've had to basically deal or witness what they have had to go through. So there are several ways in which we can treat cancer. Obviously, you can have it surgically removed if it's in a way that the doctor can go in and cut it out. They surgically remove the tumor. The other way is through radiation. And radiation, basically what that does is it destroys basically the DNA in the cell, so it can't continue to replicate if it doesn't have DNA to replicate. And then chemotherapy is another treatment that you're going to read about in the handout that I'm going to give that goes along with this video. So in apoptosis, we also talked about previously, remember, programmed cell death. Cells are programmed to basically self-destruct to allow for the formation of our fingers and toes and things like that. So know that simply cancer is uncontrolled cell growth. There's been something to basically cause the signal to basically not get shut off as it should be by the regulatory proteins. So mitosis, in summary, the big thing is it's the division of the nucleus in a eukaryotic cell, and basically they create two identical cells. They end up with the same number of chromosomes. So if you started off with a parent cell that had 16 chromosomes, the two new daughter cells are going to have 16 chromosomes. And it's only one nuclear division. The nucleus is only going to divide once. And because it ends up with two new cells, this is also a type of asexual reproduction. So what type of cells undergo mitosis? These are our somatic cells. And why do they undergo mitosis? Remember, we already talked about this. Obviously, for growth, anytime our cells need to repair, if you get a cut, the cells are going to need to make new cells to repair basically the damage that was caused from that cut, or maybe you broke a bone, etc. So mitosis, division of the nucleus, and it creates two daughter cells that are identical to one another. And we know that there are different cells in our body that are different than somatic cells. We talked about those in Unit 4. Those are our gametes, and we're going to talk about cell division of gametes in the next video. So thanks. Have a great day. Bye.